So Kenneth is professor of oncology and pharmacology and molecular science at the Johns Hopkins University School of Medicine. Kenneth received twice the award of the American Cancer Society Clinical Research. He's involved in research to define the tumor microenvironment of prostate cancer metastasis, as well as developing new therapies for prostate cancer. And as an excellent evolutionary ecologist, he will present this morning a talk entitled Applying Optimal Foraging Theory to Understand Cancer Metastasis. Thank you.
Thank you so much. It's a true honor to be here. Disclosures. So I thought I'd start with this picture of the Moffat giants, as I call them. I thought Bob was going to be here, and I don't know how you say kiss rear end in French, but I was just trying to honor this group of gentlemen who have really, I think, influenced my work and the work of the entire evolutionary ecology field over the last couple of decades, and they've certainly inspired much of what I do. I mean, the reason we're here today is that, again, metastatic cancer remains incurable, and it continues to be cancer is a leading cause of death, causing 10 million deaths last year, and about 1 in 6. And as we wipe out infectious disease and poverty and other causes of what kills young people, cancer is only going to continue to be more and more of a problem. It's so fascinating to me, especially sitting or standing in this place where Madame Curie did all of her work, that when you think about metastasis, which is the lethal event of cancer, because if a cancer didn't spread, we could just cut it out or radiate it and we would cure it, which is what happens 50% of the time now. But what we understand about metastasis has not fundamentally changed since 1889, when Stephen Paget, the great breast surgeon, wrote his 1889 paper, The Distribution of Secondary Growths in Cancer of the Breast, where he analyzed 735 fatal cases of breast cancer and argued about that the distribution of metastasis could not be due to chance. He recognized that metastatic patterns did not simply follow blood flow distribution. He referred to cancer cells as seeds, and how they distributed throughout the body must be understood by studying the properties of the soil of the secondary organs. And his famous quote, when a plant goes to seed, its seeds are carried in all directions, but they only live and grow and fall on congenial soil. And there we've sat or stood for 130 years. Working with Russ Teichman, my dental colleague at the University of Michigan, we clearly, understood that the congenial soil for breast and prostate cancer is bone. And through his work, Russ's work, and many others' work, we defined that prostate cancer cells mimic hematopoietic stem cells to home to bone, using stromal-derived factor 1 as the congenial part of the soil that's resulting in prostate cancer cells setting up in the bone. But they were circulating everywhere. So while we can define bone as the congenial soil, why do cancer cells leave the primary in the first place? And in fact, I always like to show this circuit because we forget that it's just like the bone isn't going, cancer cell, come here, I'm over here, because there's no gradient. A cell that leaves a primary organ like the breast or prostate or any solid tumor has to go through lymphatics or veins or nerves, but always ends up in the venous blood supply, which then goes through the heart, through the lungs, back through the heart, into the arterial blood supply to hit a target organ. That dissemination is agnostic. It doesn't matter how it happens, it's happening. And once a cancer cell is going by in the bone and says, oh, here's a local gradient of stromal-derived factor. I'm going to get there. I'm going to go there, and I'm going to live and be a happy metastasis. But why did it leave in the first place? What is controlling that event? And so what Joel and others have taught us is that just because you can move does not mean you must move. We have a choice. We have legs, but we don't have to move as we do things. And so Joel introduced us to think about optimal foraging theory, where in ecology there's a foraging strategy that's dependent on resources, predation risk, and movement ability. And as Joel was very fond of saying, if I'm a squirrel living in the forest, and I have enough food, and I have a warm nest, and I have a mate, and there's no fox around, I'm never gonna leave. Something has to change to make me want to go. So we've applied that thought to cancer. And as we know, everyone in this audience knows, as cancer grows, it creates a new ecosystem. And of course, that's the ecological principle of niche construction. And Sarah Ammond, a talented postdoc in my lab who studied ecology as an undergrad, taught me the word eutrophication. I have no idea what the French word for that is. I won't even try. But what eutrophication is it's when, for example, you're farming and using too much chemicals, too much fertilizer, that goes into the lake and causes an overgrowth of algae, which reduces the ability of sun to get to the grass and other parts of the system. You get decomposition and death because you have low oxygen. And that term sets up a new system. It creates a swamp. So we have actually termed this, Don Coffey, my mentor, termed this in cancer as autoeutrophication, that the decreased resources and hostile environment that's caused by cancer cells is they actually overgrow their vasculature and cause hypoxia, low pH and low nutrients, is actually creating what we call the cancer swamp. Some would call it the cancer desert, but it's the idea that we've changed the environment, that cancer itself is changing its own, creating a different ecosystem that then selects for superclones or emigrants, basically causes the species to change. So how do species react to changing environments? And this is, again, I had to put Joel's picture up because, again, we're talking on the phone and we're talking about this, and he says, well, locusts. So a grasshopper is not a hungry beast. It's eating and it is fine. But the minute that they run out of food, the grasshoppers actually transmogrify, right? And an epigenetic event, they turn into, the next generation is a locust that can fly away to find a new place to eat. I want to emphasize that that's not a genetic event. Nothing in the grasshopper's genome mutated for that to happen. And in fact, if you look on your right, you see a epithelial cancer cell, a grasshopper. And then below it, you see a mesenchymal cancer cell that's undergone an EMT, epithelial to mesenchymal transition, the locust of cancer. Epigenetically, that's a PC3 epithelial cell, a PC3 mesenchymal cell. There is no mutation there. It's already mutated, but epigenetic control. So we have actually asked, the other thing that sort of stuck in our mind is when you think about the social science concept of diaspora, where you have migrants that leave because of potato famine push, it's sort of a common anthropologic and sociologic idea that if you're a migrant, only the hardiest survive to find a congenial home. You have to have special characteristics. So we've been asking, does a stressed environment induce a migratory cell or even a resistant cell? And to do that, we've worked with Bob Austin, a physicist at Princeton, with his talented grad student Casey Lin, and my talented postdoc from Spain, Gozala Torga. And what we did was we actually created basically an ecosystem on a chip, where we could follow cells over multiple generations and multiple days without ever taking them off the microscope, and allowing us to create environments that are varied, as well as to track every cell, thousands of cells over time, to see what happens to them. And I'm not going to spend time talking about how we make this chip, but one of the key things for us was that we could create gradients of therapy. We could actually cause cultural eutrophication by putting in chemo, and as you can see with this fluorescein, we can go from high concentrations to low concentrations and hold that gradient for days on end. We can do it with pH, oxygen, chemotherapy, etc. And so we asked the question, can we watch how chemotherapy leads to an adaptation and emergence of resistant cells? And I'm going to show you a couple movies, but in this experiment, I'm co-culturing epithelial cancer cells and mesenchymal cells. The mesenchymal ones are in green, the epithelial ones are in red, and we're going to put them on up against a gradient of chemotherapy starting at 10 nanomolar docetaxel, which is a microtubule inhibitor, and at 10 nanomolar, it's basically a lethal dose for 99% of cancer cells if you just pour it into a tissue culture. And we tracked them over 2 weeks. So I'm going to see how to show a movie here. Okay, so what is going to happen here, and I'm just going to let this loop so we don't lose it, is here's this microenvironment with these colored cells, and there's a gradient of chemotherapy going this way. And what you see over time is that in the high doses, we kill everything off, but then at the end of the experiment, you see a bloom of cancer cells come back. So a resistant bloom has occurred right there. That's growing in 10 nanomolar docetaxel. Everything should be dead right there, and it's growing. So that made us think about, well, what's happening here? And I'm going to blow up that area, and there we go, and I'm going to play this again. Now, what you see here is you're going to see the cancer cells die off, but then over time, you start to see these large, very motile cancer cells come along in this area where they should all be dead. And then, towards the end of the experiment, all of a sudden you'll see a bloom of these tiny cancer cells repopulate the area. So these guys are living in 10 nanomolar docetaxel, and then all of a sudden a resistant population is born. So this concept of what's happening here really blew us away, and it was really the fact that we had this technology to watch these cells develop over time. We had this is nothing that's happening here isn't happening in cell culture, in a static cell culture flask. You just don't have the ability to look at it. So what this device did, what this little ecosystem did, was show us the existence and importance of these large intermediate cells, and then this bloom of resistant population. So we asked the question, what were the importance of these large multinucleated polyploid cells that were so motile in this move towards resistance and metastasis. So, to make a long story short, because I could talk about this for hours, we've found these polyploid giant cancer cells that are highly motile, that we think of as the locusts of cancer. They're also highly resistant. And based on the video, we thought, well, they're repopulating a non-multinucleated population that is now resistant. So we've dissected that a little bit, and the other thing I just wanted to point out is, well, are these, what this has let us ask the question, are these the cells that are actually the important cells of metastasis? We did a calculation, and this is published, so I won't spend a lot of time on it, but if you draw the blood of a patient with cancer and look for a circulating tumor cell, you may find one cell, and that doesn't seem like a lot, but you did that randomly. And if you look at, well, your heart beats 72 times in a minute, you actually put your whole cardiac output, your entire blood supply through your system every minute. So that's 5 liters of blood. And if you happen to draw one 5 milliliter blood sample and see one cell in there, that means there's 5,000 circulating at any one time. And if they clear with first pass kinetics, which most cancer biologists believe they do, that means a tumor, or your collection of tumor deposits, is putting 7 million cells into the bloodstream every day, or 2 billion a year. And if you do the math over time, essentially the ability to successfully develop a clinical metastasis in the lifetime of a patient is 1 to 10 to the billion, or it's a lot of cells. It's very inefficient. So we're asking the question now: are these polyploid cells actually the hardy immigrants? We also know now that it doesn't matter what chemotherapy or what type of cell type, these cells are develop in a dose-dependent chemotherapy to chemotherapy. So the more chemotherapy you use, as Carla was pointing out, the more chemotherapy, the dose dense, the maximal dose, the more you select for these. So that's really fascinating. We also know that we've caught them asymmetrically dividing and generating these 2N cells. That bloom you saw in the video, they're actually going through popping these 2N cells out. We also know that if you start to look for these in human tumors, they're there. But in fact, if you look in a primary cancer, a primary tumor that you may have taken out with surgery, it's very hard to find them. But the more you treat, and this is from a patient who underwent an autopsy of a lung metastasis from prostate cancer, they actually are a significant portion of the population. So the more you treat, the more you see these in vivo. Well, we didn't discover, it turns out, we didn't discover these. These are old news. Polyploid giant cells have been reported in the literature to form as a result of chemotherapy, radiotherapy, the tumor microenvironment. But it was always assumed that these things were not going to survive, that they were due to a mitotic catastrophe and were going to either senesce or apoptose. So there is a literature, it's about 20 papers in the literature, that says these are important, but nobody quite knew why. We have found in our device that they form through at least two different mechanisms. They can either go through aborted cytokinesis and form into multinucleated cells, large cells, or they can actually fuse, and cell fusion can occur. And we've documented both events actually live time. I had more movies, but from time I thought I wouldn't show them to you. And pursuant to earlier questions about stem cells, it turns out that these are stem-like. They, if you do look for a panel of STEMI genes in these polyploid cells, They are highly enriched in c-Myc, nanog, OCT4, SOX2, etc. So they asymmetrically divide. They have stem-like characteristics. They're probably stem cells. Haven't proven it definitively yet, but we're gonna. But what's also really fascinating is that now that we knew they were there, we went back and looked at our static tissue culture. And this is not the red and green epithelial mesenchymal cells. Everything in red here is a cell that's less than 80 microns in size. And everything in green is something that's greater than 80 microns in size. And this is the start of the experiment. And what you see is that these giant cells are present in 1 to 5% of every stable tissue culture population. I guarantee you, if you go back to your lab and look in your flask, they're there. Your eye is trained to ignore them, or the way we do experiments, we never see them because we just stain with a viable stain and run it through a counter and go, oh, you never see what's big or small. But they're there, and they're selected for as you with your stress. The other thing that's really fascinating, and this is with Lindcamp cells, is that okay, we can flow a population of cells and determine what percent are these cells. So if you have in this case, a Lindcap population of cells that sit at they're already a little bit aneuploid, so they are 2N plus. If you look at what percent of the population at rest that's not stressed, it's 3%. You treat them with, again, a lethal dose of docetaxel. 3 days later, they're 90% of the population. And then you remove that stress, and 3 weeks later, they're back to 3%. The exact same percentage you started with, but now you also have a resistant population of 2N cells. So they've birthed, somehow, this resistant population, but they're back to 3%. And I can't get over that. I can't get over that. There's somehow, they know they have to be in the population somehow. There's some community sensing. And so, I don't know anything, but the idea of, I'm thinking about this now in terms of potentially, can we think about this as a cancer hive? Inside a hive are multiple types of bees that have specific functions. The queen bee, the workers, the foragers, the cleaners, the drones, the undertakers, the nurses, the builders, the temperature controllers, and the guards. They actually do this because through pheromones and age. Like a 1-year-old bee one day old bee has a different function than an 8-day-old bee. They actually, the older they are, the less work they have to do. They age out of having to do hard work. But the number of them, and if the hive is injured, all changes based on pheromones secreted by the collective. So right now we're thinking about, well, if we think about as pheromones as chemokines or cytokines that are being secreted by the tumor ecosystem, should we be thinking about tumors as superorganisms? An old concept where you define the superorganism as a collection of agents which can act in concert to produce phenomenon governed by the collective, and there's no brain here. I'm done. Thank you. So our current experiment is to ask, how does conditioned media control the percent of queen PGCCs in the tissue culture collective? I'll skip the philosophy and say thank you.
Thank you very much, Kenneth.